Welcome everyone to our webinar on MicroRNA-4553P. My name is MD Ariful Islam. I am a postdoctoral research fellow in the Reddy Lab at Texas Tech University Health Science Center, Lubbock, Texas. Today we are going to discuss about the role of MicroRNA-4553P as a peripheral biomarker and therapeutic target for mild cognitive impairment and Alzheimer's disease. To begin with, let me explain how miRNA works. MicroRNAs are small non-coding RNAs that regulates gene expression. MicroRNAs are transcribed in the nucleus by RNA polymerases as primary microRNA, which is then converted to precursor microRNA by DROSA and DGCR8. Precursor microRNA is transported to cytoplasm by X protein 5 and RNA-GTP complex, and then cleaved by dicer and TRBP to a final double-stranded microRNA. Later, it is incorporated to Argonaute complex and form a miRNA-induced RNA silencing complex. In this complex, microRNA interacts with 3' untranslated region of target mRNA to induce RNA degradation and translational repression. MicroRNA-455 is highly conserved in most of the species from chordata to mammalia. This microRNA is transcribed from human collagen type 27 alpha-1 gene. MicroRNA-455 has two different isoforms, MicroRNA-4553P and MicroRNA-4555P. MicroRNA-455-3P has key roles in different types of cancer, cartilage development, adipogenesis, preeclampsia, amyotrophic lateral sclerosis, and Alzheimer's disease. In today's presentation, we will focus on the role of MicroRNA-455-3P in Alzheimer's disease. In our lab, we investigated the role of MicroRNA-455-3P in Alzheimer's disease in two different models, in vitro AD cell culture models and in vivo transgenic and knockout mice models. In the later part of our presentation, we will elaborate how MicroRNA-455-3P is linked with the Alzheimer's disease pathogenesis and development. In the beginning, MicroRNA-455-3P target prediction analysis showed amyloid beta precursor protein as a putative target, and luciferase reporter assay confirmed the interaction of MicroRNA-455-3P at the 3' untranslated region in the APP gene. In N2A neuroblastoma cell lines, overexpression of MicroRNA-455-3P resulted in significant decrease in APP in a RT-PCR assay. Conversely, MicroRNA-455-3P inhibitor treatment increased APP level. Furthermore, MicroRNA-455-3P treatment also significantly decreased the mutant APP expression. As we know, amyloid beta precursor protein is processed by alpha secretase, beta secretase, and gamma secretase enzymes. MicroRNA-455-3P initially targets 3' untranslated region of APP gene and decrease the level of APP gene. It also negatively regulates beta secretase enzyme BACA1, and ultimately there is a decline in amyloid beta peptide accumulation. Next, we studied the role of MicroRNA-455-3P in mitochondrial biogenesis in a cell culture model, where overexpression of mutant APP decreased the protein levels of PGC1 alpha, NRF1, NRF2, and TFAM, which was rescued when co-expressed with MicroRNA-455-3P. This data indicates protective role of MicroRNA-455-3P in mutant APP-induced mitochondrial dysfunction in Alzheimer's disease. In a similar fashion, MicroRNA-455-3P showed protection against the deleterious effects of mutant APP in mitochondrial dynamics. MicroRNA-455-3P decreases the expression of mitochondrial fission proteins DRP1 and FIS1 and increases the mitochondrial fusion protein UPA1, MFN1 and MFN2 when co-expressed with mutant APP. This data indicates that microRNA-455-3P rescued mitochondrial dynamics in Alzheimer's disease. The role of microRNA-455-3P in mitochondrial health and function was further supported with our electron microscopy data. Here, mitochondrial number was decreased and mitochondrial length was increased after microRNA-455-3P overexpression. And it also rescued the effect of mutant APP treatment. Furthermore, we reported that microRNA-455-3P prevented mutant APP-induced synaptic dysfunction. MicroRNA-455-3P overexpression rescued the protein level of synaptophysin and PHD95 when co-expressed with mutant APP in N2A neuroblastoma cell lines. To sum up, in our in vitro cell culture model, microRNA-455-3P targets 3' UTR of APP and reduce mutant APP levels. Furthermore, microRNA-455-3P rescued mutant APP-induced mitochondrial dysfunction and also prevented mutant APP-induced synaptic dysfunctions. In our lab, we generated microRNA-455-3P transgenic and knockout mice model. Transgenic mice lived 5 months longer than wild type. It has improved cognitive behavior, spatial learning and memory. It showed enhanced spine density and synaptic activity with improved mitochondrial biogenesis. Whereas microRNA-455-3P KO mice lived 4 months shorter than the wild type. It showed cognitive impairment with mitochondrial and synaptic dysfunctions. In our Morris water maze experiment, microRNA-455-3P transgenic mice showed superior performance relative to wild type and knockout mice, whereas the wild type mice performed better than knockout mice, which indicates that depletion of microRNA-455-3P impairs memory and cognitive functions in mice. As we have discussed the protective role of microRNA-455-3P in mitochondrial function, microRNA-455-TG mice showed similar results. MicroRNA-455-TG mice showed increased mitochondrial biogenesis protein, PGC1 alpha, NRF1, NRF2 and TFM, and it also showed increased mitochondrial fusion proteins, UPA1, MFN1 and MFN2. And it showed a decrease in mitochondrial fission proteins DRP1 and FIS1. PGC1 alpha, NRF1, NRF2 and TFM, these proteins are associated with mitochondrial biogenesis machinery and associated signaling process. Here in our Western blot analysis, we collected protein lysate from mice brain cortex and found a significant increase for these proteins in microRNA-455-3P TG mice compared to control wild type mice, whereas microRNA-455-3P knockout mice showed reduced expression. Next, we analyzed the protein expression related to mitochondrial dynamics. Mitochondrial fission proteins like DRP1 and FIS1 showed a considerable decrease in microRNA-455-3P TG mice compared to wild type mice, whereas in microRNA-455-3P KO mice, expression for DRP1 and FIS1 slightly increased. In case of fusion protein, MFN1 expression were significantly increased in TG mice compared to the wild type mice, and knockout mice showed a deep decline. To understand the impact of microRNA-455-3P on synaptic and dendritic activities, we also assessed the levels of synaptic proteins in wild-type TG and KO mice. The densitometry analysis revealed a significantly increased levels of SNAP25, PH95, and MAP2 proteins in TG mice compared to wild-type mice, whereas knockout mice showed reduced level of expression. These indicates microRNA-455-3P improved dendritic health and synaptic function. Further, we conducted Golgi-Cox staining in the hippocampus of 12-month-old wild-type, transgenic and knockout mice. We found that total dendritic length and spine density were significantly increased in transgenic mice compared to wild-type and knockout mice. And knockout mice showed a significant decline in both the parameters. These observations suggest that microRNA-455-3P overexpressions improves memory and cognition in TG mice by improving neuronal health and spine density. To support our previous findings, we also performed electron microscopy to analyze mitochondrial number, mitochondrial length, and synaptic density in cortex and hippocampus of transgenic wild-type and knockout mice. It is shown that healthy mitochondrial morphology prevails in TG mice compared to the wild-type, and spine density is significantly increased in transgenic mice compared to the knockout and wild-type mice. To sum up, from our investigation on microRNA-455-3P transgenic and knockout mice models, we found that overexpression of microRNA-455-3P improves mitochondrial function, synaptic density, memory and cognition, and extends overall life expectancy. For the first time, we reported elevated microRNA-455-3P level in serum, cerebrospinal fluids, AD postmortem brains, and cerebral cortex. We also found upregulated microRNA-455-3P in APP transgenic mice and different AD cell lines, for example, AD fibroblast and AT beta lymphocytes. We suggest microRNA-455-3P as potential peripheral biomarker for Alzheimer. Next, we designed a kit for analyzing microRNA-455-3P from blood for early diagnosis of mild cognitive impairment and Alzheimer's. Overall, 20 ml blood will be drawn. RNA will be isolated from the serum to perform polyadenylation and synthesize cDNA using microRNA-455-3P specific primers. Fluorescence spectra will be used to confirm microRNA-455-3P levels. In this method, we can diagnose the risk for Alzheimer's in a non-invasive and peripheral manner that will help to take preventive measures to delay or escape Alzheimer's disease development in later age. To investigate and establish the superior role of microRNA-455-3P in the prevention of Alzheimer's disease, we are planning for genetic crossbreeding of microRNA-455-3P TG mice with different AD model mice, namely APPKI, 5XFAD, and mutant tau mice. We will examine different AD-related features like amyloid beta toxicity, neuronal damage, mitochondria and synaptic features, and overall memory and cognitive functions in these crossbreeding mice. In a similar fashion, genetic crossbreeding of microRNA-455-3P knockout mice with AD model mice will be employed to find how depletion of microRNA-455-3P can be linked with the development of Alzheimer's disease. To conclude, our take-home message is microRNA-455-3P dramatically decreases full-length APP expression, the C-terminal fragment of APP, and reduces amyloid beta levels. MicroRNA-455-3P mice models showed its vibrant role in maintaining and promoting memory and cognitive functions in an extended life period. MicroRNA-455 serves as an ideal biomarker for diagnosing mild cognitive impairment and Alzheimer's disease progression. We highly appreciate your time and interest in our webinars. Thank you.